All right. Welcome everyone. We have a tight agenda, so we'll get started on time here. Welcome everyone to the final presentation from the RECCS 2019 cohort. Yeah. And so I really want to welcome all the students. I want to welcome the mentors, and I know some mentors are going to come later. That's why we put a program together. So thanks for everyone who's here yet, and to those that are going to come later. Welcome also to some of our advisors and previous RECCS team members, Leslie and Patrick here. And then there are some peer mentors. So Jessica, the one I've seen, yeah. Anyway, and then of course the RECCS team. There's a lot of us supporting, but especially Jenny and Renee have been doing a phenomenal job this summer. So I'm just really excited to be here. I'm Anna Gold. I'm the director of Series Education Outreach, and I'm running the RECCS program with this great team. And yeah, it's the 6th summer of RECCS this year. I just did the numbers this morning. We had 61 RECCS students. It was really fun at the poster session yesterday to actually see about 10 of our alums come and support the students. And you all had peer mentors this summer, so it's really fun to see the RECCS family merge together. And you're all going to be part of the RECCS family now, and hope to continue being engaged. So it's been a full summer. It's crazy to think about this. Like 9 weeks ago we started, and you all didn't know anything about activity or like photogrammetry or whatever your topics are. I know it was hard in the beginning, and look at where you are. You did a fantastic job all of you today. And I know there's been lots of ups and downs for all of us, just like sciences. There's been a lot of challenges, a lot of successes, but then also, times where you were like not sure you were ever going to be in this room and presenting. So look at you all. We've seen you all present on Wednesday. You're going to do really well today. I don't want to take too much time here, but I just wanted to talk just a little bit about who makes this happen here. So first of all, obviously, if we didn't have the 13 students here, this wouldn't be a program. So thank you all for coming in with such a phenomenal attitude and just really working hard, all of you. We ask them in weekly surveys about challenges of the week and what they learn, how many hours they put in, we chart them. And it's sometimes crazy to see these charts like going well past 40 and putting in weekend hours. And I know you all work really hard. Then of course the mentors. This wouldn't be possible without the mentors. And thank you all for being supportive, encouraging, maybe sometimes frustrated how much time it takes, because we're all short in time, but I hope you're all proud together with your students where you got here. And then of course it takes people to run a program. Renee has put a lot of time in. You've gotten like a lot of emails from friends and bolted and very well formatted, so you could all follow along emails from her every week. So thank you so much to making the program run really successfully. And then Jenny Briggs has run in the 2nd year now the science communication workshop, and you've all heard from her at the beginning of the program. I think it wouldn't be without Jenny that you all have these amazing posters and really had a lot of critical questions. What's the axis here? What's your question there? Can you do this plot a little bit better? So I think Jenny has done a great job guiding you all to get here. And then we have photographers. Keith, yes. Thanks a lot. We have our tech and IT team is not here, but they have supported because a lot of RECCS students went to Alaska, went to Gothic, went to, I can't remember, Wyoming. So you all had to Skype in at different times for your presentations, and thank you to the tech team. And then we have support students, like Anna has been helping, and some other students in the other room. So lots of people helping. And of course, it wouldn't be RECCS without funding. So thanks to all the funders. We have funding from the National Science Foundation. There might be some program officers on the line. So thanks, NSF. We have support through different programs through the RU site. We have funding through the LTR site grant. And then we have some additional funding through supplemental grants. So that's supporting all the students here. Anyway, this is also say welcome to series. We are really excited you're here, and we can't wait for the posters. And in the program, this is a tight schedule here, and then there's going to be a celebration in the end, so please stay for cupcakes later on. We are excited to get started here. The students have been doing great, but there's a lot of, what did you say, Kat? You said confident nervousness. So there's a lot of confident nervousness in this room, which I think is an awesome agreement. But I just wanted to talk to the audience. We all, there's this rumor in the world that academic settings are really rigorous. And yesterday we actually had some very rigorous reviewers of posters and some very friendly ones. But it would be fantastic if in this room we could keep it to constructive questions, keep asking questions of the students, but please keep them positive and encouraging and kind of celebrating, because this is a celebration of the summer. And anyway, with all of that, I'm excited for the first presentation.
And as our first presenter gets ready, I want to encourage friends and family to ask questions as well. You don't have to be a professor or a graduate student to have an insightful question about the projects the students did, so please feel free. And we're going to have 7 minutes for each student's talk, and then 3 minutes for the question session. So we'll have a timekeeping team here helping us stay on track. All right, our first presenter is Nelson Hyder-Kuhn, who worked with his mentor, RECCS, and colleagues at Ceres and NOAA. Thanks, Nelson.
Thank you. Good afternoon, everyone. So my project is When North Goes South, investigation of selective geomagnetic variations in Boulder, Colorado. So what is geomagnetism? Geomagnetism is the study of the Earth's magnetic field. Humans use geomagnetism in a few ways, mostly for secondary navigation protocols, as well as high-tech navigation and directional drilling in oil and gas. In my project, we use what we call our CrowdMag app. It's an application that's been developed by NOAA. What we do is tap into the magnetic feature of collected data to collect data, and then we use that to map local anthropogenic features as well as geologic features. In our project, we ran into what's called magnetic anomalies. These are localized deviations in the Earth's magnetic field. So just keep that in mind and for our future slides. Our goals were to increase confidence in CrowdMag measurements, as well as compare different modes of transportation, i.e. walking, biking, and car travel in our data. Questions that we had for our project this summer was how far can we trust maps from CrowdMag data, as well as does the mode of transport affect the data? And then lastly, what can we do to improve data reliability? Long-term goals of our project is to create models of Earth's time-changing magnetic field by combining crowdsourced magnetic data with data collected by ships, aircraft, and satellites, as well mapping local magnetic noise sources, i.e. from transformers, pipes, lines that are in the ground, etc. Our methods section, we went to 2 locations. We did repeat measurements in different modes of transportation at each location. We used multiple phones, primarily iPhones, and then again, we did multiple transportation modes in our surveys. After we got the data, we put all of our data into Google Sheets. We removed any abnormal shifts in the data, as well as corrected the amplitude just to really compare all of the data sets that we gathered over our surveys. And then after that, we took our final residual data set and then removed the residuals so we could see third order polynomial fits or long wavelengths that we could compare to Earth's geomagnetic field. Through this we can interpret identification of zones with distinctive variation patterns, as well as compare the reliability of our data compared to Earth's geomagnetic field. For our first survey, we did a walking survey, a biking survey, and a vehicle survey on 4th Street in Boulder. As you can tell, we have a lot of pretty scattered residuals in these 3 columns. That's our that's as clean as we could make the data for this. Something that we noticed was how individual parked cars definitely affected all of our data noise that we gathered. So that was really hard to split out. There was some repeatability of vehicle data, but overall it disagreed with our walking data trends. However, our walking and bike data trends, our polynomial data trends, actually matched pretty nicely with the geomagnetic field on the very top. For our second survey, we went to Williams Village. We could not drive a vehicle at that simply because we walked on sidewalks, probably frowned upon if we drove vehicles on sidewalks here. So yeah, we stuck to walking and bicycling, but what we found was the data really matches pretty similar with each of the surveys that we took in both modes of transportation, which was really nice to see how clean data could actually be. Then we did what we called our Krav Mag Day. Krav Mag Day is when we involve people from our intern cohort. We had 5 walkers with this, and what we did is mapped an entire area rather than just a linear outline of an area. This was really cool because we had never done this before, and so what we did is we took the data points in the second graphic. We used a GeoSoft program to map the magnetic variations, and we found that we had actually mapped the basement before we even knew that the basement was there. We actually laid down engineering and construction models in this photograph, and unfortunately, I included a larger picture, but it is not on my slide now. But yeah, it was a really cool thing to see there. So with conclusions, with the data collected from user surveys of this experiment, we can tell that with proper observation and data processing, our Krav Mag application is fairly accurate. By determining which mode of transportation a citizen scientist is using, we can interpolate the data appropriately, efficiently, and effectively. We have also determined that a data survey can be significantly affected by geomagnetic noise in the surrounding area created by cars, light poles, underground plumbing, etc., any metals that are in the area. And then additionally, our multi-user mapping effort on Krav Mag Day successfully demonstrated the ability to map an area which had never been done before. This may be relevant to many future mapping applications for our Krav Mag user teams. What's next? As far as the application go, development of better calibration procedures in the Krav Mag application, as well as the ability to designate and separate modes of transportation for better assessment of accuracy of data is definitely an important thing. Understanding larger anomalies in the Krav Mag data stream by conducting additional ground truth exercises, like our full mapping of that area, and then further development in our Krav Mag flight mode, which the team just got funding for. It's going to be used during commercial airlining flights to start mapping that type of transport. Personal acknowledgements. Thank you for the RECCS team. I wanted to thank my mentor, Rick Saltis, and the Geomag team at NOAA, as well as my husband, who couldn't be here today, and my recommender. She also could not arrive today, but definitely a lot of inspiration there. And then, as we always say, map before, share the data.
But we actually have about 4 minutes. Go ahead, Kate.
That was really interesting. Thank you for sharing.
Yeah.
I was wondering, so your results showing that the biking and the car, the biking and walking in the car results were inconsistent, made me wonder how often does Krav Mag calculate, record magnetic data, and sort of how does that relate to the speed that the person is moving? So like, what's the observation frequency, and I would also wonder, in thinking, we speculate about how what that might mean for existing airplanes.
Yeah, absolutely. So in the application itself, we can set the data collection to approximately 3 seconds. Now we have noticed different variations per different phone, as well as like the type of upgrade a phone is. So sometimes we'll set it to 3 seconds, but it'll be like between 2 and 3 seconds. And then 3 seconds for my phone is 3 and 4 seconds. So we had to, in our data interpretation, we actually mapped the general meters that we had and tried to interpolate the data so it was all in the same playing field in that sense. So that's the best that we could do as far as taking that time consideration with how you map data to the slides very quick comparatively. But that's still in development. So yeah, absolutely.
You said that the biking and the walking was somewhat close to each other, but the driving was different.
Yeah.
If I heard correctly.
Yeah.
And with that, where were you holding the phone when you're riding the bike?
Yeah. So we actually put it in our shirt pocket. We removed our belts, so we wouldn't have any effect there. And we, on our Williams Village, we actually rented those boulder bikes. As far as the comparison with a vehicle, I definitely have to say that, you know, when you think of a vehicle, it's made mostly of metal. And so it automatically is producing magnetic variations just by itself, which was apparent in our first survey. So there's definitely, it had never been researched before. It had been speculated that that may affect the data, but now with observing different modes of transportation, we can at least separate all of that data to interpret it better. Yeah, absolutely.
Nelson, I have a quick question, because I'm not sure you mentioned where you did that multi-user survey, under which there was a secret basement.
Yeah, so we did that at the Duane Lawn Physics building. It has a huge lawn right in front of it, and we were pointed in that direction because we knew that there would exist some type of classrooms or something under it, but we didn't know where or what. And so that was a great way to explore doing an entire area map of something that we were unfamiliar with. And it was actually really awesome. My mentor and I got hundreds of emails from the engineering and construction crew trying to show us all different like electrical lines and plumbing lines, but the actual map that we both decided and agreed on actually worked really nicely. And the basement is literally pretty perfectly centered under that magnetic variation that all of our phones picked up. And so that was pretty awesome.
Cool. All right. Any other quick questions? Thank you. That was really cool. All right. Our next speaker is Keely Lawrence. And as you may know, we have several different disciplines represented here. So all of our speakers come from different subfields, and we try to encourage sort of common language and themes, but we're changing topic now to geology. Keely worked with Katie Barnhart and Greg Tucker here at CU in the geology department. Thank you, Keely.
Hi, thank you. Hello. Okay, so the title of my project is Mapping an Active Debris Flow Site Using a Deep Convolutional Neural Network. What is a debris flow? It's going to be a surge of colluvium and water going down a hill slope with whatever it carries. They are usually triggered by a rainstorm event and can be really destructive to anything at the bottom, whether it be people or property. On a larger scope of this project, colluvium, which is sediment and rocks, is what feeds the debris flow. And examining colluvium coverage changes, sorry.
That for you. That sound good?
What? Okay, examining how colluvium coverage changes in the debris flow site and would allow us to monitor possible initiation conditions efficiently at possibly like higher risk sites. So where are we going to study this? The Chalk Cliffs Basin in Buena Vista, Colorado is one of the most active debris flow sites in the state. Its high frequency of debris flow initiation makes it a great outdoor laboratory for studying debris flows. Which leads us to the next question. How are we going to map surface features like colluvium? If you are not familiar with deep convolutional neural networks, or DCNN, okay, because I was not either of them. They're a class of computer-based learning algorithms, which are pretty effective at image recognition and classification. So why would we use a DCNN for mapping the basin? We have a database of over 10,000 high-resolution drone images of Chalk Cliffs Basin in our database. Classifying them by hand can take up to an hour each. DCNN could provide a more efficient means of doing this for us. So the question we set out to answer this summer was, can we in fact train a DCNN to classify surface features? And if so, how specifications to the training set could contribute to the resulting accuracy? The first thing we set out to do was build a training set. I selected 114 drone captured images from the database, semi-randomly to display a variation between surface features. All the images that were chosen were taken between the summer of 2017 and the summer of 2018. Next, the raw images were cut into quarters, and 61 of these quarters were uploaded to an annotation software. For the DCNN to learn, it first has to be told what was what. So we classified the content of each image into either tree, bedrock, or colluvium. From that, we got the image and its corresponding annotated layer, which we cut into smaller tiles, and those tiles were randomly assigned to either be a training tile or a validation tile. And what this means is when the DCNN is training, it's going to look at the training tiles and my annotations, and then it's going to test itself on the validation tile and give itself an accuracy score, or an AVA, average validation accuracy. Once run, we ran it 3 times total in 3 different training trials. And here are the results. So on the x-axis, we have the number of epochs, which is the iterations of training refining the DCNN. On the y-axis is the average validation accuracy, or AVA, which is the score it gave itself and how well it did. So as you can see here, it's divided into the 3 classes, but for sake of time, we're just going to look at the total. The biggest things we can notice from this graph is that the number of epochs after about 150 epochs, variation in AVA begins to lessen. There we go. Okay, and the accuracy between trials 1 and 2 is greater. Sorry. There's better accuracy in trials 2 and 3 than in 1. And the main difference in this was we reduced the size of training set only to include the most thoroughly annotated images. So from the study, we can conclude 150 to 200 epochs is an ideal length for the training period for our study. Consistency and annotation of images may produce a higher AVA. Training can improve with a better quality training set rather than a larger quantity. And finally, with a 70% average accuracy, the DCNN can be trained to identify surface features. And I'd like to acknowledge the following. My mentors and Jess and Frank for recommending me, and the rest staff.
Thanks so much, Keely. Any questions? Go ahead.
So could you remind me, what was the best accuracy you got from your algorithm?
For the total AVA, it was 0.698, so about 70%.
And how accurate is a typical person?
Like a typical, like running it with an object training?
Yeah,
Or
let's just say you took an image and classified it.
well, it's not objective because it's what I think colluvium is and bedrock is. So that, I mean, would be 100% accuracy on my part because the DCNN, what it's learning is how I would classify bedrock, colluvium, and trees, not necessarily what it is.
I don't want to dominate the question.
No,
You're, between, so if you did classification and then your mentor did a classification and another scientist did a classification, how similar are they going to be?
We didn't do that because we tried to eliminate any variables like that because of the fact that it is going off what I'm deciding is colluvium. And there were already variations in how I was annotating throughout the 7 weeks that I was annotating images overall. So if we could do it again, I would re-annotate everything with one consistent level of annotation and just me doing it.
Maybe to add to that, how long would it take if 3 different scientists did their own thing? I was sort of trying to follow Rick's question.
For annotating
Would it be
images?
more or less efficient than this network automation process?
Ooh, well, it would be less efficient for the total purpose. Because, I mean, we have 10,000 images in the database, and for like the big picture of the study to track colluvium over time, you would have to, you would have to annotate 10,000 images and then put that on a time span to see how colluvium coverage is changing, and that would be a lot. I mean, that would be 2 years, just 10,000 images. And if you wanted to do it a longer period of time, you'd need a lot more images than that. So yes, this would be the most efficient method.
Any other questions or volunteers to annotate thousands of images? Yeah, Claire.
Can you use the training sets that you pioneered in this way to identify the diagraph between change and sites?
Ideally, yes, and potentially, if there was more time, I had, I think, 400 images from another debris flow basin in Switzerland with similar composition and like looks to the colluvium here. So the next step would have been to run those through without annotating any images and see if the validation accuracy matched up to that 70%. And that would be what I considered like a success in that matter. And then from there, yeah, you could potentially put more in, and that would be the goal, to see if to see if it could be used to be applied to different sites. Yeah.
I assume one of those might be.
That's me, and that's Jess, and that's
So did you actually then do some ground truthing to make sure that you were accurate in how you were identifying the different renders?
Can you rephrase that?
Were you out in the field, did you ground truth, like make sure that, what you were assigning colluvium was correct and the rock and tree?
Yeah. So yeah, we were out there, and it was definitely easier to see being there from after looking at hundreds and hundreds of pictures and being like, yeah, that is colluvium right there. And otherwise, before that, and I did most of my annotations after being at chalk cliffs, but being able to zoom in really far on the pictures and just use your sense of, okay, well, this is a sharp hillside. There's not going to be colluvium right here. And then you can see where it piles up. Anywhere also, it's pretty telling if there's trees. It's usually in a colluvium field.
Well, thanks so much. Great questions. I'm going to try and switch speakers right on the dot. Our next speaker is Alex Brunson, because some people have to come in and out to watch certain talks based on their schedules. So continuing a geoscience or earth science theme, we have,
It's getting there. 400 slides.
oh, no, I love she's kidding. We have Alex Brunson, who worked with Carly Brooker and Ben Littleday and team. Thanks, Alex.
All right. Hello, everybody. We worked on quantifying burn severity through laboratory wildfire simulation, the dirt on wildfire. So wildfires are an increasing problem in the US, and fire intensity generally refers to the above ground effects. So vegetation burning, faunal recolonization, structural restoration, etc. However, there is a call to define the difference between fire intensity and burn severity, which refers to above and below ground ecological effects. And specifically in this study, we focused on soil burn severity, so mostly the below ground ecological effects. And different wildfires have different severities. So we wanted to put this into a quantitative metric instead of a qualitative low, medium, high. So our objective was to use a quantitative temperature-based scale to replicate moderate, low, and high severity wildfires through laboratory simulation. And this shows the different varying temperatures and its effect on soils and plants at different temperatures in degrees Celsius. So our methods section, this was a huge part of our project. I'll try to skim over this because I'll go back to it later. But we collected soil samples from the from the location of the Hyde Park fire in 2012 in the Cache-la-Poudre Basin. And we transported these soils and we covered them in plastic because a really important part was to maintain the integrity of our soil samples in these little soil boxes here. And we also took soil moisture profiles of the area, triangulated it, and then dug in that area. We then put them onto a cart, recorded the date, time, air, the ambient air temperature, the weight of the soil moisture profile, and placed them under a 375 watt bulb, as you can see. And this is our fire simulator that we had to engineer from scratch in order to try to replicate wildfires. We then inserted thermocouples into these little holes that we had drilled into the sides of our soil sample boxes so that we could measure the temperature. And thermocouples are these long temperature rods that read really high intensity heat. And we have a little thermal logger here, which also records data in the temperature time curve. And as we recorded and as we lit things on fire, we took that data and we had a lot of different variables that we looked at. So we were trying to achieve different temperatures. So we classified it as low, moderate, and high, these different temperature grades. We looked at the temperature time curve, so the time it took to reach the maximum temperature and the fall. Thermocouple placement was important. We used the same, kind of the same ideas as Cancelo Gonzalez, who is a fire god, and I refer to him often and look through all of his research papers, and he was definitely a strong influence on making our experimental design. And we looked at a lot of different variables for simulation, or for the simulation as well. We looked at here's an example of it. It's anaerobic combustion. But like I said, we looked at the soil moisture, the initial weight. We looked at the distance of the soil from the bulb because we thought that this might also influence the maximum temperature we're able to reach. And we compared all of these variables against one another to reach the maximum temperature. They take about an hour to 2 hours a piece. So in total, out of 59, I only got data from 33 of them, and it was hot data. Here we can see that our maximum temperature versus the time to reach maximum temperature is all over the place. It shows a lot of variability, illustrating that there are a lot of sources of variation that we were not sure how to account for. So here we thought that soil moisture would be like the #1 factor affecting the maximum temperature reached. But as you can see, I split these into our moderate temperatures versus our high severity. We had about 22 moderate severity burns and only 8 high severity burns. But as you can see, like the initial soil moisture at 5% and 11% only reached a maximum of about 450 degrees Celsius. So somewhere in there, there's some other variability. Same with the duration to reach our max temperatures. So what could this be? Qualitatively, we did awesome. Once again, comparing to Cancelo Gonzalez, our time temperature curves definitely match that of someone who has a lot of research in this area. And qualitatively, we have the similar look as the BEAR assessment, the Burn Area Emergency Response Team, for a moderate wildfire. As you can see, the spatial variability are the 2 bulbs, so there's some variability there. But what we didn't account for was wind and wind factor and wind mitigation. So as you can see here, it looked like a really nice time temperature curve, and then it gets a little crazy, and that's because the wind started to blow. So what we've concluded is that we need to do this study with wind as a controllable factor because that definitely influenced our data. And wildfires in themselves, though, have a spatial variability and a spatial randomness. But I believe that with more controlled simulations and perhaps putting all of the same soil moistures in one area or having a wind tunnel or a wind control, that would definitely have a controlled variable that affected our maximum temperatures. But we were able to reach things qualitatively and emulate low, medium, and high severity wildfires overall. So we were successful in that. And as wildfires continue to increase throughout the western US and are a problem, and I guess in Alaska as well, it's valuable to further our research in this. And I think this really sets a nice premise for further research in quantifying wildfires because they are an increasing problem. And I would like to thank everybody at the RECCS program, our grants for funding my second job and experience. And unfortunately, my mentors could not be here today, but I'm very grateful and to the staff and the members of the research. So, yeah. And once again, Consuelo Gonzalez.
So good job, by the way. The sample was taken from an unburned section of the Hyde Park fire. Is that
yes. So what we wanted to do, this is a smaller scale part of a larger study. So we wanted to compare soil samples to previously burned samples of the Hyde Park fire because there was a lot of research done on post-fire sedimentation and debris runoff from the Hyde Park fire to the Cache-la-Poudre Basin. So we're literally, as you can see here, this is actually one of our sampling sites, and they're doing a prescribed burn. But in the background is the border of where the Hyde Park fire ended. So that way we kind of have a basis to base our soil sampling off of the severities that they have taken from the fire itself. And they also have done a lot of research on the soil profiles from the Hyde Park fire and how that affects sedimentation. So we have a basis of comparison.
Is there a possibility that had an impact on the burn area for the Hyde Park fire, and that may have actually affected your sample?
I believe there may be some impact from the fire. We do know that where we were testing was unaffected by the fire. It was just outside where the fire kind of ended. I do not have the data for the soil profiles from the Hyde Park fire, literally at 20 yards from where we were sampling from.
Sorry, I'm late. Have you faded?
So, because this was so experimental and we engineered this, we initially were like, okay, we'll just do a little, and we'll do a little.
I think the door was closed trying to revive it now.
Because there are so many variables, we weren't able to control.
Not right now. Okay.
I mean, we could control things by turning on the bulb.
Well, because I'm not sure we might.
Turning on the bulb, the temperature would drop immediately. But what we thought was more interesting was tracking the maximum temperatures we.
Izzy is getting ready to present her research from the summer. That's what I've got on video there. She'll be on in a minute.
Control the temps to desired temperature. Right. Well, sorry, we will need to switch to the next speaker, but say
the live feed with the thing that is. She's not.
Please mute your mic.
Now, for people who may just be joining us, at the end of our talks around 3:30, we will have a reception with cupcakes and drinks next door, and you'll be able to ask any of the distinguished speakers more about their study. But our next presenter right now is And Kat worked with Ellie Brown's group here at Series chemistry department.
Just tried doing some.
So my project was an analysis.
Is Sadie at this place right now?
Not Sadie, Izzy.
Izzy. Oh, I was gonna say.
Aerosols are small particles suspended in the atmosphere, and they can influence the climate in two ways, by direct interactions with radiation and indirect interactions by affecting cloud formation. And both of these interactions can cause both cooling and warming or warming. And we have biogenic aerosols, or natural sources, those come from volcanoes, forests, salt. Anthropogenic aerosols, those are human created. They can come from agriculture, power plants, cars. So radiative forcing. Radiative forcing is the difference between the sunlight that's being absorbed by the earth and the energy that's being radiated back out. And if you look at the x-axis here, above zero, forces temperatures to rise, zero forces temperatures to fall. In aerosols represent the highest uncertainty in anthropogenic radiative forcing, and you can compare that with CO2, which is one of the biggest drivers. And relative to the aerosols, that's a very small error bar. You see the large error bars for the aerosols, that blue box there. This uncertainty here is believed to be in large part due to the uncertainty surrounding aerosols. So that's why aerosols is important. So my research used aerosol data collected as part of the 2-column aerosol project of TCAP, and that was collected February through March in 2013. With the aerosol mass spectrometer, which separates particles by size and provides info on chemical composition. This figure is from the TCAP science plan. This first column is the average optical depth, or the thickness, and the red is representing high concentrations. Second column is average radiative forcing. The blue is representing cooling effects. The third is the standard deviation of the models. And you can see from the high amount of the difference between the first and the third column that there's a high amount of uncertainty in this area. So my project focused on determining origin and transport of aerosols. In order to do this, I had to organize large amounts of data for different experiments, different time periods, different collection intervals, and large gaps in the data. I used this, I did this using Igor Pro, and I learned to code with Igor Pro, having a prior experience in coding. And I wrote from my own code, and I created all my graphs with this software. On the x-axis here, we have February 4th through March 6th of 2013. On the y-axis, we have micrograms of aerosol per cubic meter. That's our absolute aerosol mass loading. And the aerosols we're looking at with this data are chlorides, ammonium, nitrates, organics, and sulfates. I also looked at black carbon, and you can see when overlaid on the aerosol measurements, there's definitely a relationship there, and I will get back to that. So the first thing I looked at was these two sections on my graph, the high spike and the low point. I wanted to compare where the air was coming from for these two areas. And so I did this by running air mass back trajectories, and I used a model called HighSplit, and the data for HighSplit is directly available from the HighSplit database. So it's measuring the air that's coming from down here going up to Cape Cod, and we found that for this high point, the air is
chocolate crackers.
We have New York, Jersey, urban environments. And then over here, the low point, we have air coming in over the ocean, marine environments. And these trajectories were run through a 24-hour period every 4
Well,
hours.
are you dressed? Annabelle, I want you to go ahead and get dressed, okay?
And
Because
then
we're,
to get a better look at the composition.
I'll tell you what, why don't you go ahead and get dressed for me first, and then you may have to.
Sections.
Okay.
She
wants the
chloride
chocolate crackers.
that is coming over the ocean.
then
Over to
I
Amos.
did all of this fractional data by wind direction, and I came up with this. This is bulk aerosol measurements from different regions. The charts are sized based on mass loading. Mass loading region. So we have the largest from the southwest, smallest from the southeast, and you can see that the largest, it actually doubles in comparison to everything from this region, and from the smallest one here, it actually triples. So that's quite a big difference. So what's going on in these regions? We have forest and rural up in the northwest. We have ocean to the southeast and more urban areas to the southwest. And what's interesting and surprising about this is that the mass loading differs quite a bit, but the actual composition within each pie chart, it stays very much the same. So I concluded from this that urban areas are the most influential factor for increased aerosol mass loading. There's a lot more sources for aerosols coming from urban environments. And the association between the black carbon and the aerosol measurements indicate that an important source of aerosol during this time is combustion. And again, it's surprising that the mass loading differs by region, but there's no significant change in the composition, as you can see from the largest mass loading versus the smallest mass loading. The composition is very much the same. There's a bit of an enhancement of the chlorides coming in from the ocean. And with that, I'll take any questions.
Super interesting. One of your earliest slides, I think you showed the effects of aerosols on the radiator balance. And did it show that the
To be able to see Izzy, but I think Izzy's getting ready to start.
effect than the other aerosols? Did I read that right?
Boom. Is this already doing it?
Or is that just an error bar? Your upper bar within the aerosol and
To the right?
yeah.
Yeah, that would be the black carbon.
Okay, so that gives a positive forcing.
Yes.
And the other aerosols give a negative.
Yes, that's correct.
Yeah, that's super interesting.
Yeah. Go ahead.
You said something about all the other directions except for the board.
Yeah, that would be more ocean, and there's.
That's enough. Go ahead and turn off the TV and hop in the car. Okay.
Yeah, I wasn't super concerned with that direction. More so the southeast and the southwest. I'm not 100% sure what exactly is in that.
Pause it, you turn it off.
We will.
So we're going to continue our atmospheric theme here with our next speaker.
Oh, there's Izzy.
Elizabeth Izzy
Here, hold it.
They have 3 affiliations: Ceres, NOAA, and GMD. Oh, great. We have a lot of acronyms on our sheet. But go ahead, Izzy. Thank you.
Great, thank you. So my project is a study of ambient levels of air toxics near a multi-well pad in a residential neighborhood. So oil and natural gas production has been increasing rapidly since 2010, and this is largely due to the implementation of fracking and directional drilling.
Mom, you want me to get in the car?
The population expansion in has meant that are.
I want you to run inside with my phone and keep it on, okay?
So previous studies in Denver Basin and Northeast Colorado have found elevated mixing ratios of volatile organic compounds, or VOCs, and methane associated with oil and natural gas extraction. Methane is a greenhouse gas that is affecting climate change. VOCs and NOx in the presence of sunlight create surface ozone, which is a respiratory irritant. It's like sandpaper in your lungs. So for this reason, EPA has regulated this to 70 ppm. And the Colorado Front Range has gone over this level every year since 2004. is a subgroup of VOC, which is benzene, toluene, benzene, and xylene. And these are highly toxic. They can cause everything from eye irritation to cancer. So our Broomfield campaign was from July 2nd to July 27th this year. And we deployed 2 instruments in a mobile trailer near a multi-well pad. And I will be analyzing a 10-day period. So our Broomfield site, we have our lab about 167 meters away, 16 well pad in a residential neighborhood. And you can see we have a road going just north of our site, and this is important to me because VTEX is also emitted by vehicles. So we will look at the relationship between VTEX and other compounds to differentiate the other local sources. So to do this kind of cost-benefit analysis, we need 2 instruments, a gas chromatograph, which is measuring our VTEX, and a ring down cavity spectrometer, which is measuring carbon dioxide, carbon monoxide, methane, and water. So the VTEX instrument, when it gives you a measurement, it's a chromatogram. And each of these peaks represents a different compound. And the area under the peaks is the concentration. So you can see that sometimes it's not actually measuring the right area, or it's measuring, or it's not detecting the peak at all. And this means you have to go through and manually correct each chromatogram to measure the correct area. And from our preliminary data before this process was done, you can see that our references and our calibrations were all over the place, and that you can actually see a daily cycle in the references, and this indicates that we were having some temperature issues. But once I went through and corrected 1,902 chromatograms, we have much better looking data. So I'm comparing 4 different sites, our Broomfield measurements against the Boulder Atmospheric Observatory in Bella Romero, which are also measurements near oil and natural gas operations, and Niwot Ridge, which is our regional backgrounds. So you can see that we did get some maximum levels well above Niwot Ridge, but overall they were lower than in Bella Romero last summer. There's 2 reasons that this could be the case. Our wind speed and direction for the 10-day analysis period, and mostly the winds are coming from the north and the south. And since our site is west of the oil pad, they were really only concerned with winds coming from the east. And we have there 2 wells were drilled in this time period, and the drilling mud was changed from well 1 and well 2 to a synthetic based that doesn't have any hydrocarbons in it. But we did see 2 elevated readings in each point for each well period. And you can see that the highest plumes are coming from well 1. So focusing on this plume, I saw a relationship between methane, toluene, and benzene. They were likely from either co-located or the same source.
Here you go.
Max level of 5.28 ppm of methane says that this is a pretty close source. And our winds for just this analysis period, just when the plume was, are coming from the east, as you can see from the other windrows. And this tells us that this relationship between methane and benzene and toluene, that if for well 1, the benzene and toluene is in the drilling mud, but everybody see any methane there. So that's certainly not the source. And the
15 kids today.
The CO says the emission sources from vehicles. So one hypothesis that we have.
Is she in her pajamas?
Well, I woke
when they were drilling
Stop it.
the
Stop.
zone, so the source rock that has the oil and gas in it, either before or during they started horizontal drilling.
Oh, she gotta say something a lot.
So to summarize, comparatively, we are seeing
Oh, really?
higher levels of hydrocarbons than in other studies, but
This is better composed and more comfortable in front of me. I'm gonna get her in a minute. I put her at
could be coming from the oil and gas.
Be nice, you all. It's been a terrible day.
Still had development operations.
Hi, sweetheart. Hi.
With
Hi. I'm so
well-designed,
happy to see
high-quality
you.
measurements, we can support.
Can I have a kiss? Can I have a kiss too? Can I have a kiss?
Next steps are to collect measurements for a longer duration, more and have a more robust attribution to sources, and to collect measurements during other
Oh, okay.
drilling or
Honey, come over here. You watch it too. You wanted to see.
to compare to ambient levels.
I can't get the app on my phone.
And
Maybe without the
everybody
Is that it?
possibly.
I mean, I went to Zoom.
everybody else who works with Noah.
And the internet work is so sorry that I couldn't get it to download. So I left.
Ryan, get to be there.
Thanks so much, Izzy. Any questions?
I just asked her this morning, when was it going to be? And did she want a new dress or something to wear for it? Yeah, no, she never answered me. And then I got this to say, she never answers me.
I'm not sure of that one. I heard that Colorado State has its own.
Hi, baby.
And I think that's set at 9 ppc per hour.
Could bring that thing. Could you?
Yes. I'm not sure. I think our plume only lasted for like 45 minutes.
Kevin's coming home tonight. Oh, to get the dog and go back to Colorado. He's in Chicago. He was waiting to try and get a trip south to pay for the gas, but he's getting the dog and heading back to Colorado.
I personally
that he
think
loves
I know
Colorado.
that
He says Ollie's gonna love it here.
yeah,
Oh, look at this. God, she's the cutest kid. Oh, honey, I wish you could have seen her face from over here.
Did she just kiss you?
The baby, not me. Did you see Izzy?
Yeah.
Yeah, she watched it all the way over here.
Oh, did she?
Yeah. So I was like, we get to Alma's. I want you to run that inside.
I don't know.
Yeah.
But we ran into issues with and so there's things out before and having air conditioners a little bit better so that we don't have to reintegrate all of those
Types of challenges.
and maybe
Can I have a Ortiz now?
You can have whatever you want.
All right, well, thanks so much.
He's done. Really don't watch the rest of it.
Thank you all so much for your attention. I feel like it's a mini seminar here. We have Austin presenting also in the general field of atmospheric science. And after the break, we will switch gears to ecology topics and also sort of interdisciplinary studies and snow. So Austin Ortiz worked with John Albers and Juliana Dietz at Series, NOAA, and ESRL. Thanks, Austin.
Thanks, Jenny. So hello, everyone. Glad to see you all here. So the title of my project is The Effects of the El Nino Southern Oscillation on Stratosphere to Troposphere Transport over the Western United States. But before I get really into the project, I'd like to give a little bit of our motivations and goals. So ozone is a naturally occurring chemical in the upper atmosphere and stratosphere. As Izzy pointed out very well, ozone is very bad for your health, and especially down here on ground level, the lower atmosphere, the troposphere. However, it can be produced locally due to factory emissions, local pollution, things like that. But during the spring, it can be periodically transported down to the troposphere due to large wave-breaking events. So being able to predict the timing and strength of stratosphere to troposphere ozone transport may be very beneficial to ozone for air quality forecasters. But right now, we know that the location and strength of weather systems are known to be related to the location of the jet stream. So our hypothesis going into this was that the El Nino Southern Oscillation is known to cause systematic shifts in the jet. So we might be able to harness that knowledge to predict the timing and the location of really strong stratosphere to troposphere ozone transport over the western United States. But before I go any further, I'd like to talk a little about ENSO and about what it is and what it does. It's 3 to 7 years, and it's sea surface temperature oscillation over the central and eastern tropical Pacific Ocean, and that causes east-west shifts in convection, rainfall formation, and also shifts in that convection will then cause north-south shifts in the jet. And so it comes in 2 cycles. El Nino, which causes the jet to be further south, as you can see here in this top part of this schematic given by NOAA, and La Nina, where the Pacific jet is further north, as you can see in the bottom part of this schematic here. Going into this, we had an interesting methodology. We used a couple of different data sets, one from the European Center of Mid-Range Forecasting, and another from the ETH Zurich ERA interim. And that we used the European data set for wind data and the ETH for mass transport data. With that, we created a climatology, which is basically just the average of all of that data, because what that is that it's an expected normal climate. It's something that we're really not interested in. We really wanted to look for anomalies. So once we had that climatology, we subtracted it from all of the data to get us more values that swayed outside of the normal. If you're interested in the math of that and how the data was collected, there's a small bit of what we did. After we did that, we had to calculate when the ENSO periods were. And with that, we used a data set from NOAA that looked at sea surface temperature oscillation. We weren't really interested in the data as much as we were when the ENSO events were taking place. So anything below one standard, or above 1 standard deviation was considered to be El Nino, and anything below negative one standard deviation of the ENSO index was considered to be La Nina. Once we had those times, we created composite plots using those times. We created 3 different things. We created wind plots at 200 hectopascals, which is at the tropopause, which is the boundary between the stratosphere and the troposphere, which is known that we can, which is known where we can see the jet stream as well as the storm track. We also looked at 500 hectopascals, which is the it's the mid-latitude troposphere, and the planetary boundary layer, which is the surface of the Earth. I wanted to take a look at, this is the 200 hectopascal wind data. The gray composite here is the actual jet stream. This is a normal jet stream during the springtime. So this is what we would expect it to look like during just any kind of springtime locations. This is wind frequency data. I don't really want to get into it too much, but if you just think of this region as positive and this region as negative, and you add and subtract it from our climatology of the jet, you'll see that the jet will actually start pointing south for El Nino, and the opposite's true for La Nina. If you add these 2 together and subtract this, you'll find that the jet stream moves further north. With that out of the way, just wanted to kind of let you guys know what the jet stream was doing at the time. This was the actual mass transport data. As you can see here, the positive mass transport takes place in the southern part of the United States and the upper part of Mexico, and corresponds very well with our position of the Pacific jet during that time. The next one is at the planetary boundary layer. As you can see, it's the northern part of Mexico, really southern part of the United States, exactly where we would hope it would be. Corresponds pretty directly with the 200 hectopascal data of the jet stream at the time. For La Nina, you can really see where the jet is here. You can see that the polar jet shifts further north, which corresponds where we would hope it to be. So this is really good in supporting our hypothesis further. For the planetary boundary layer, you can see here, it's in the southern part of, the northern part of the United States. It's above Mexico, and it really is a great support for our hypothesis. So what does it all mean? Well, it means that stratosphere to troposphere transport of mass is directly affected by the El Nino Southern Oscillation. It also shows another thing in our results. It shows that higher air quality can be expected during El Nino periods due to the amount or lack thereof of this transport, and you can expect lower air quality during La Nina because of the amount of transport that is taking place. Hopefully, these results will lead to improved air quality forecasting over the Western United States during the spring and early summer. I'd like to take a minute and just personally thank Renee, Jenny, and Anna for this amazing opportunity, the National Science Foundation, and a resounding thank you to my mentors, John Albert and Juliana Diaz, whose constant support has really led to this. Thank you very much.
Thanks so much, Austin. Who has questions?
Is there any measurement of how much enhancement there is at the ground level from the transport?
I'm sorry, how much what?
How much enhancement, how much additional ozone there is at the ground due to the transport?
So that's probably,
Measurements that, or
yeah, I'm sure that there is measurements like that. My mentors are going to be doing that for future work. Right now, we're just looking at just mass transport. We can see that mass transport, there's quite a bit taking place in La Nina, not quite as much as in La Nino. Excuse me, El Nino. So really, that's all we can really determine right now.
There was a lot of details to absorb there, but one part of your processing, you did some work to decide whether you were in an El Nino or a La Nina
Yes.
event, and I imagine that others have also worked to characterize or to classify. Did you compare your classifications with possible other researchers who sought to say whether for sure we're in one or the other event?
So this was such a complicated release scenario to get through. So we kind of just did our research and kind of called it. We understood that data, it was sea surface temperature oscillation. We understood that if it was greater than what the standard deviation would be, then that's definitely an El Nino. And if it was less than a negative one standard deviation, then it's definitely La Nina. So we just kind of took that and ran with it. And the results were great. They showed it pretty well. They showed the shift in the jet exactly where we hoped they would. So.
And again, we might be able to get some supplemental input from mentor groups or other epiphanies and follow-ups. But any other questions? I'm kind of curious, following your question, as to whether now that we've absorbed a little bit of this, if we hear such and such is going to be an El Nino or a La Nina year, would you say you have some evidence that there could be more or less ozone problems, or is it a little too early to make those predictions on the ground?
So so our second result, is basically, so during El Nino, there's a lack of that mass transport, so you're definitely going to get a better air quality year. For La Nina, this is relative to the Western United States, of course, but during a La Nina, you're going to be getting more mass transport, which is worse air quality. So, this is just transport from the stratosphere to the troposphere, and that includes ozone. How much, that's for my mentors for future work for them.
So there's a silver lining to El Nino. Sometimes there's big flooding or torrential rain,
and that's due to the change.
The air will be fresher for us.
Absolutely.
All right, well, any quick follow-ups? Maybe time to give you all a break, and we'll reconvene in 10 minutes at 2:20. Thanks again.
titled Environmental Factors That Influence Nest Box Utilization by Black-capped and mountain chickadees. So birds provide many services for us, specifically ecological services such as pollination, seed dispersal, pest control, which I'm very thankful for, and they're very important in many food webs. So we are specifically comparing black-capped and mountain chickadees. Black-capped, Peaceolea atricopilus, prefer lower elevations and mixed forest and riparian zones, while mountain chickadees, Peaceolea gambelli, prefer higher elevations, although they are a little more varied in their preferences, and non-deciduous forests. And morphological differences, the black-capped looks like he's wearing a little cap, the top of his head's all black, and the mountain chickadee kind of looks like a little bandit with his mask over his eyes. How you can distinguish between the two species. So my project goal was to evaluate environmental factors that affect nest box utilization. So the three factors that I specifically looked at was the aspect, so how the nest box was hung on the tree, the elevation, so there were three locations, Boulder, including CU's campus, Sugarloaf, and the Mountain Research Station. And the third factor affecting utilization was the tree type, so either deciduous or non-deciduous trees. So the methodology that was put in place prior to me starting with this lab, but again, nest boxes were allocated at those three locations. They were placed on either deciduous or non-deciduous trees, as mentioned, and the nest boxes during roughly the period from April through July, they were checked 2 to 3 times per week. April through July is roughly the breeding period for both these species. So nest box utilization is defined as the formation of a nest, and clutch initiation, which is the laying of the first egg. This is our site study map. So you have Boulder starting here, Sugarloaf is in the mid-range, and then the Mountain Research Station is the highest elevation. So the first graph that we're looking at here on the x-axis is the tree type, and on the y-axis is the count of nest boxes. So again, black-caps prefer deciduous trees, while mountain chickadees prefer non-deciduous. This is nest box utilization by aspect. So again, on the x-axis, we're looking at the type of aspect, pardon me. And then on the y-axis, we're looking at the nest box count. So both species had a preference for southeast facing aspects. And this graph is by elevation. So on the x-axis, you have a range of elevations. Black-capped had a pretty strong preference for lower elevation, while mountain chickadees varied a little bit more than black-capped, but still preferred higher elevations. The nest box count is on the y-axis. So out of the 335 nest boxes, 84 were utilized, 38 were by both species. Black-caps utilized 24 of those 38, and mountain chickadees utilized 14 of those 38. So in conclusion, just to recap, deciduous trees were utilized, those nest boxes were utilized more by black-caps, and the non-deciduous by mountain chickadees. And then the southeast facing aspect was utilized predominantly by both species. And then black-capped and mountain chickadees utilized nest boxes at similar rates in elevation ranges between 1,700 and 2,000 meters, which is a significant pattern. This pattern shows a potential for a hybridization zone. Hybridization zone, a hybrid, an example would be a mule. That would be a cross between a donkey and a horse. Thank you. Sorry. A donkey and a horse. So the black caps and mountain chickadees, they're very closely related, but as I mentioned, there are distinctions. But we are seeing that in that zone that is circled in red, the rate that they're using is similar. So again, that pattern could indicate that there is a hybridization zone taking place. More research and further field seasons are needed for that. So this is a nest box. These are the nest boxes that were used for our project. And then I do have my little emotional support.
Which species is that?
This is a black cat. It's not true to size.
Any questions for Vanessa?
Yeah, in that table a couple of slides before, it looked like you had a high number of songbirds in that like first yellow.
Yes.
Row. Are they out competing a lot for nest boxes? Did you find that problem?
Well, this was our first official field season, so yes, that did happen. So those other passerine birds were house wrens and nuthatches. So they have similarities to black cap and mountain chickadees as well.
Go ahead.
I'm just curious, is there a reason why, well, are nest boxes needed? Are they not endangered, but I mean?
And nest boxes, they will form nests in natural like tree cavities, but we wanted to examine them, and we did capture them, and we had measurements that we took. And so, and especially for that hybridization, to test that, we do have to do a DNA extraction.
I'm trying to do the math in my head, but maybe you know the answer. I was trying to see at what elevation range that you had the highest utilization, recognizing the fact that you have to samples.
So the highest was between the range of 3,200 to 3,300 meters, and that was a mountain chickadee.
For the highest elevation?
Oh, that.
No, I meant where you had more of the nest box being utilized by either type of chickadee.
So
of the available boxes.
So the black caps definitely have a preference for lower elevation. So that's right here. That was 20 out of 24 black cap nests.
I guess my point would be that there were 65 boxes that weren't utilized at that elevation as well.
Yes.
And so if we were to basically take a look at, could it be that there were more using those boxes at that elevation simply because there were more available?
Well, and that we definitely saw some of that, again, with it being the first field season. I mean, the same is true of southeast facing aspect. Most of the boxes were put up at southeast facing aspect, but overall they still showed a preference for that. So I definitely think that we need to do more research, and we are planning on putting up more nest boxes, and I think that will even out some of the counts in the varying ranges.
And also, people might be able to participate in this study because I think it's connected to a citizen science project.
It is.
So ask the team more about that. All right, thank you. Our next speaker is Claire Atkins. Come on up, Claire. And she will take us even higher into the mountain zone, the alpine zone, looking at pika ecology. Claire worked on this study with her mentors Chris Ray and Ashley Whipple of eBio and Instar and the LTER. So thank you, Claire.
Thank you, Jen. So the title of my project is Trouble in the High Seas. This is the influence of thermally stable refugia on Rocky Mountain pika occupancy. What? Microrefugia are intersections of habitat that offer shelter to plants and animals during periods of environmental adversity. They're species dependent and also regionally dependent. So one place that provides refuge for a species in this part of the country might look totally different for the same species in a different part of the country. Pika tend to occupy high elevation rock slopes that look like this. This is an example of a talus. And so the purpose of this study was to determine which thermal factors are influencing the persistence of American pika at Niwot Ridge, Colorado. So pika are seen as an indicator species for climate change for the following reasons. They are extremely thermally sensitive. Their high metabolic rate keeps them within a really narrow thermal range of being able to survive. So they're subject to both heat and cold stress mortality. They practice limited dispersal, which means that once they establish territory as adults, they will not leave in search of more suitable or better habitat. So when they go, they're gone for good. And they're also easy to detect. So if you like hiking, you might have heard this. At any point. They also stack their fecal pellets into little mounds, and they spend the entire summer gathering forbs and grasses into hay piles, which we'll look at in the next slide. So part of the methods for the study were looking at occupancy status in both 2016 and 2018. We identified occupied sites as those which had fresh pika sign. So this is an example of a fresh hay pile, and these are examples of freshly stacked fecal pellets. And then old or no sign were unoccupied sites. So we used 2 surveys that had been were taken prior to my involvement in this study to look at how occupancy changed over time. These are study sites that I visited. So this is all Niwot Ridge, Colorado, and then up here we have the Green Lakes Valley, 15 total. So using temperature data, there were subsurface sensors that were placed into talus to capture the conditions which a pika would experience. From 2016 to 2018, these sensors picked up temperature every 1 to 3 hours, and then I separated that raw data set by summer months and winter months. We then used a logistic regression of 2018 occupancy status on a range of temperature metrics which were informed by prior research that indicate thermal factors of microrefugia and or drivers and predictors of pika occupancy and extirpation across the western United States. This is an example of coupled versus decoupling in habitats. The results of this were that in 2018, occupied sites experienced less variation in temperature between years. So of the statistics that we ran, the most significant, the model with the best support included 2016 occupancy and stability of maximum temperatures between summer 2017 and summer 2018. So here, on average, occupied sites experience less variation of extreme temperature in the summer than unoccupied sites. What does this mean? So these results indicate that Niwot Ridge pika are utilizing microhabitats which provide stable and long-term refuge from summer heat. But this is interesting because the drivers of occupancy and extirpation in different parts of the western United States are inconsistent. So while their utilization of various habitats is impressive, it is ultimately limited by the availability of that habitat. So pika tend to occupy places that are characterized by permafrost and subsurface ice, and because the physical aspects of their habitat aren't being degraded in the same ways, a lot of local extinctions are driven by climatic factors alone. So loss of permafrost means loss of refugia. It doesn't matter how good they are at locating places to hide, they're going to run out someday. These are the professional acknowledgements that I would like to personally thank the RECCS staff and cohort, Chris Ray, Ashley Whipple, and then also Don Cummings for recommending this program.
All right, some questions for Claire. Yes, sir.
Do you think we could make, or you could make little pica boxes like? Could you make them?
Well, yes, the interesting thing about that is because we can't really determine what is going to make up a good habitat for them because it's such a wide range. In other parts of the country, they're occupying like low elevation boulder fields that are covered in moss because that acts as a great stabilizer of temperature. They live in lava fields in other parts of the United States. So it's sort of hard to predict what kind of habitat we would build for a pika, and it would be really, really specific. But in the future, using fine-scale microclimatic thermal data from surface level and then being able to overlay other abiotic factors like elevation, aspect, all of those things, we might be able to map out where we could find suitable refugia for pika as a future contingency.
Other questions? I'm noting that Rick, as a geoscientist, is asking a lot of questions, so I'd like to call in some ecologists. Yeah. I guess I'm curious, Claire, you did some field work this summer. Can you tell us a little bit about how that related back to the 2018 and 2016 surveys?
So I visited most of the sites that I had temperature and occupancy data for, but the snow melt this year was such that I didn't get into the field until July. So my use of that occupancy data was limited by that inconsistency.
Did you see them? Did you see signs that they were in many of the same sites from the previous years, or was it really variable?
It was pretty variable. What was interesting is that I spent most of June just like diving into this data set, so I got to know the sites themselves by their temperature patterns. And when I would go out in the field, I'd be like, well, this one has, you know, it's experiencing this like huge range of temperatures and it's not stable at all. And then I would go and it'd be fully pika. So the prior research aspect of this was really interesting for me, at least, because it informed the hypotheses and the models that I ran. And most of them, what is cool is that a lot of the limiting factors for populations in other parts of the country don't seem to be affecting Niwot Ridge pikas in the same way.
That's good news. Any more questions? Yeah, Elder.
Do you know if there's a lot of data in other regions for pika occupancy and temperature, so that you can compare the two data sets and see is this area maybe buffered from different kind of effects regards to maybe those other regions? Have you looked at that at all, or like maybe that's from from the next bit of the work?
Yeah. So every metric of temperature stability, or the extent that a site was decoupled from the ambient air temperature, all of the metrics that I used were from prior research. So there are a lot of different factors that could be influencing the way that pika are interacting with their habitat. Unfortunately, it is so detailed and variable that I didn't have the opportunity to set them up side by side and sort of look at it that way. I was really interested in just finding out what was statistically significant in this area using this metric.
Maybe next summer compare
Yeah,
regions.
next time.
All right, we'll change speakers now. So we're going to continue the high elevation adventures with Brandon, our next speaker. And he went to multiple high elevation sites, so has some data on what makes Niwot Ridge different from those other regions potentially too. Brendan worked with Kelly Karskadin of the eBio department and her team. Go ahead.
Thanks, Jenny. All right, so yeah, we were studying the flowering time and reproduction between parent and hybrid taxa of subalpine cinquefoils. Give you a little bit of background on this particular plant that we're looking at. It's in the rose family. It's the genus Potentilla, commonly known as shrubby cinquefoil. That kind of envelops a few different species within that genus. We were particularly looking at Potentilla hippiana and Potentilla pulcherrima and its hypothesized hybrid offspring. So with that, why were we studying the flowering time of this? Well, studying flower time kind of gives you the insight into hybridization, or gives us insight into hybridization across the Colorado Rocky Mountains and its response to future environmental change. Things such as range expansion, which is taxa moving to different environments, bringing it into contact with other species. A positive effect of this could be adaptiveness, like is the plant able to respond well to environmental change. A potential negative effect would be outcompeting other plants within the area for resources. So yeah, these are kind of some of the reasons why you want to study hybridization. Really, flowering time is the first step in the potential form of hybrids, so this is really where you want to start. So how did we do it? We had two locations, one at the Rocky Mountain Biological Laboratory, which we call Rumble, and then one up at Niwot Ridge. These are both located on one on each side of the Continental Divide. We had 3 sites at Rumble that we looked at, and then 4 sites at Niwot Ridge where we collected our data. My time with this project was about 5 weeks in the field, but for the data that we are focusing on here, it was about 2 consecutive weeks, roughly 2 to 4 days of collecting data that we're going to be looking at. What we did was we looked at flowering synchrony by counting the number of flowers per individual, and we also looked at the total potential output. So we counted the number of flowering stems, multiplied that by the average number of buds and flowers per plant, and that's how we got that number. Any differences in the taxa were analyzed with ANOVA and Tukey tests. So the stages of reproduction, Barry Brosey's lab down at Rumble put together this great little chart telling you about phenological stages that you're able to mark down. We modified it a little bit to fit our plant. So stage 0, we marked vegetative, where there were no reproductive structures being produced on the plant. Stage 1 was budding, so no flowers yet, but it's starting to produce. And then flowering, if we saw a single flower on the plant, then it was in stage 2. And then stage 3 was senescence, meaning that there are no more active reproductive structures on or being produced on the plant. So some of the results, we found overall that they were pretty well in line with each other as far as what stages they were in at the places. But what really kind of grabs my attention with this here is that we were seeing a lot more reproductive activity beginning to happen at Rumble than we were at Niwot. Why is that? Maybe did it have to do with the moisture, the latitude? We had a lot of different weather patterns between here and there. We noticed it didn't rain as much during our study at Rumble as we saw that it did at Niwot Ridge. So maybe that had something to do with it. But overall, more reproductive activity west of the Continental Divide. As far as total flowers being produced during our survey window, we saw that on the chart on the left there, the hybrid species was definitely producing much more flowers just overall throughout both of the sites, indicating a slightly earlier but overall synchronous flowering time. But as far as reproductive potential goes, the parent species Pulcherrima was producing a lot more reproductive structures during our flowering time. Now, whether or not those all made it to maturity, I didn't have the time to look at. But yeah, overall, greatest number of flowers were the hybrid, but Pulcherrima had the greatest reproductive potential. So what are we doing now? competitive advantage. Are these hybrids better at things such as like defense against predators? We saw a lot of pollen predation going on, things like that. Are they more or less adaptive? Are they hybrids able to respond to environmental change better than its parent taxa? Or are they producing viable pollen to begin with? Few preliminary tests we did were looking at the receptivity of the female parts by doing a little test with hydrogen peroxide. The bubbles there indicate that it is able to receive pollen. That was a lot of fun. Also looking at the viability through use of a staining agent. We had trouble finding mature pollen during this study, and that may have been due to predation, that may have been due to the late flowering time and just the time that I was able to work on this. But the clear dots there indicate empty pollen grains. So the hypothesis is that you should be able to get an idea of the overall viable pollen production per flower by doing these staining tests. Also, something that was really exciting was doing some in vitro germination. So testing different concentrations of sucrose to water by weight and watching the pollen tubes grow. We found that 20% sucrose to water by weight was actually the secret ingredient here. So that was fun. Next time, we'll definitely be trying more of that. And then also pollen predation. I think I would like to look at more to see what nominal levels of pollen predators are existing. So I think a lot of this should really be over the long term. I think really just a few weeks.
I'll tell you what, driving was hard.
You excited about it.
I drove home.
So overall, what did we get out of this?
They didn't recommend it, but
had overall synchronous flowering time with their parent taxa. The hybrids produced more flowers earlier.
It was tricky, but
the parent species
did it.
Had the greatest reproductive potential overall. And again, this is just the time that we really looked at it. Also, environmental conditions kind of limited. Survey a complete reproductive cycle for all three.
Luckily, I have my toes sticking out.
And then real quick, I just want to thank my field team, Bill, Andrea Clement, and of course,
Miss your shoes.
I was driving and I can stop and my boot is stuck in the brake and the stick.
Oh no. Jeez.
Yeah. Sometimes they're a lot easier to tell than others. Sometimes I need a set of eyes on it. Kelly's been looking at these for a long time, so she's a good idea. The Hippiana has innately compound leaves, so it has kind of looks like I guess like a single stem.
Oh no way.
And then the experiment has palm leaves.
It was like a restaurant?
So, like, coming off your hand.
Oh my goodness.
Hybrids can have kind of a mix of that. So you might have three.
Was anyone hurt?
That are coming off.
No, I saw in the place, it was about tables of the food.
That's really.
Oh, thank God.
Oh my goodness, yet, but that's really exciting. So finally, you know, when
That's so cool.
Jeez. Oh no. No.
So that was really interesting. So overall, these main sites, roughly 2,900 meters in elevation. But we found like Hippiana likes to grow on more regions. So we had another site at Rumble that was
Whiplash
Hill. Wow, that was pretty much all Hippiana. And we went literally across the road onto the flat ground river, and that was pretty much all Proterma. So yeah, that's a good question too. Is you know, how far?
Okay, what is that?
There are places
on my shirt.
Little
Yeah,
places.
what is that?
It's a sloth.
Why are your toes sticking out?
Thanks so much.
I don't know. My shoes in here. I took them off.
Hey, Christopher, could you please mute your mic? Oh, shit. The one police was really nice. The first ones came and said, "I need one." He said, "No, I'm not. They come another police array of the living organisms to you during this afternoon. But Elder will describe his work with Jesse Colangelo and Sebastian Kopp. Thanks, Elder. Take it away.
Thank you. All right, so this summer I looked into a bacteria called Desulfobacterium autotrophicum. Now, that's quite a bit of a mouthful. We're just going to call it D-auto for short. Now, a little bragging to begin with. This is a photo that I've taken with an SEM microscope, and to my knowledge, this is the best photo of this bacteria that exists. Okay, so vanity aside. So, desulfo or D-auto is a sulfur-reducing bacteria. So let's talk about why that's important, or what about sulfur is important. So things like the sulfur cycle, it's a very important biological system that affects the entirety of the Earth. Understanding the aspects that affect this, and bacteria are a huge mover of the chemical composition of the Earth. So it's important to understand how they interact with it. Now, the part that we're interested in particularly is this relationship between the bacteria and the mineralization or the mineral records. So in order to understand a little bit more about that, we're going to look into something called fractionation. Now, fractionation is, well, it's a measurement of isotopes, which is a difference between heavier and lighter versions of an atom. So we're going to be looking at sulfur. Now, fractionation is the difference between that ratio of heavy and light ones in a system, or heavy and light of that element on one side of the system compared to the next, or the other side. And our system will be the bacteria. So in order to get this sort of information, we're going to use something called a chemostat. Now, what a chemostat is it's a way of controlling the growth rate of our bacteria so that we can control for that factor in determining its relationship with the sulfur species. So we have a bioreactor where the bacteria grow and do their thing. And we have a reservoir that consistently feeds it more media or more food at a consistent rate, so they can control, make sure that it is growing at the rate that we want it to. On the other, so if you reflect this photo over in that direction, there's a second chemostat that we've set up. So we have two of these things running at the same time, so that we can control different rates simultaneously. So in order to pursue this information, there's two things that we need to measure. There's the metabolism of the bacteria, and then its fractionation. So to get the metabolism, we look at both ends of what it reduces, the sulfates and the sulfides. And to do that, we use ion chromatography and a sulfide assay to measure those concentrations in the media. And then we relate that to its optical density, which is a measure of how many cells are in the media at that growth rate. Then, after we collect that data, we compare it to mass spectroscopy of the sulfur species, which separates the isotopes so that we can measure that ratio and relate its fractionation to its growth rate. So, while doing this, we've discovered a few interesting things along the way, which relate to its growth and its interaction with its environment. So, this is a figure representing the first chemostat that we set up, and the blue line represents the optical density, which is the amount of cells in the media, and then the yellow line is actually a bajillion little dots of data points of pH. So it looks as though pH is very closely associated with the number of cells in the media. So what we see is when we as the cells increase, they also increase the pH. And we're not certain why it's causing the pH to change. We think that maybe it's the sulfide interacting with some part of the media, or changes the pH itself. But we're not sure at the moment. The other interesting thing is that here's the second chemostat, And you can see that that relationship between pH and optical density is a lot tighter in this chemostat. Is that the second thing we found is this bacteria seems to grow a lot slower in our chemostats than we initially expected. In little bulge tubes or in small volumes in batch culture, where they just kind of sit there and slowly eat what they've been given to start with, they've been shown to grow at a doubling rate of every 12 hours, meaning that the number of cells doubles within that time frame. However, in our chemostats, we found that that has slowed down to about fourfold, so it's 4 times slower growing in our chemostats. We're not certain why that may have been. It may have something to do with the media. Maybe there's a limiting reagent or factor in its composition. So we'll have to look into changing up that a little bit. So back to that fractionation thing so we've collected some information in regards to the metabolism of the bacteria, but we're still waiting to get some fractionation data because the lab that we previously were using is no longer accessible, so we now need to send our samples to England. So that'll take a while to get. Yes. But getting this information would be really integral to understanding more about the relationship between bacteria and the and mineral samples. So, have any questions? Yes.
So how did you isolate the bacteria to grow?
Isolation. So you mean like keeping it so it's not other bacteria in that media?
Yeah, like you look like you had a pretty pure culture.
Well, you can buy specific strains from other companies that do that for you. But another thing that's special about this bacteria is because it's a marine bacteria, it has a high salinity affinity, and so that's one thing that kind of differentiates, that keeps it from other bacteria wanting to go into its media. Is a lot of salt. That doesn't keep it isolated from all bacteria, but it does help within the lab environment that we were in. Yes.
Do you have any hypotheses about the things in the media that
I think that it may have been that there wasn't enough sulfite or sulfates in the media to begin with, and that may have been suppressing their growth rate because then that was purely dependent on the flow rate. But if there wasn't an abundance of sulfates to begin with, then it's that factor controlling their growth instead of the flow rate. And so it wouldn't matter how we change the flow rate up to the degree that it allows the bacteria to have an excess of that reagent. We did a data point this morning in regard to its metabolism, which showed that it seemed to be metabolizing the sulfates as soon as it gets them, or like within that range. So that kind of gives credence to that idea that it might be that there's a limiting region in the media. We need to change that up.
I might have time for one more short question. I'm curious who these collaborators are across the ocean.
So back there is Sebastian Kopf and Jesse here, and they helped quite a bit with guiding through this process. And where else was the other
I'm curious what connection you guys have with the lab in England.
They were willing to let us use their mass spectrometer because sulfur, a lot of different facilities don't like having sulfur in their mass specs because it's kind of sticky. Yes.
Technical term.
Yes, just well, yes. And some specs aren't set up for those compounds. The way that it's set up can limit what compounds you can put in there.
Well, interesting twist. Yeah. Thanks, Albert. All right, we're going to stick with our microorganism theme and call up Savannah Pierce as our next speaker. Savannah may have traveled the furthest in the course of the summer. You'll see where she went with her team. I'd actually like to recognize the Theory Lab. Who's here from Noah's lab? Oh, yes. They often arrive in large numbers and have been awesome RECCS mentor teams for several years. So Savannah, thank you, and take it away.
All right, so over the summer, my project that I did was culturing and sequencing novel microorganisms found in boreal mosses. So the first thing I'm going to talk about is the northern latitudes. The north has a lot of diverse areas, as you can see. It's really important for the carbon cycling and especially watershed formation in northern areas. It also has a lot of unique plants, and these plants are really sensitive to climate change because they can kind of only grow in this area, some of them at least. And if you see, if climate change actually starts to take hold in the northern latitudes, many of these plants may actually not come back. So why are mosses important in the northern latitudes? Mosses cover everything in Alaska, everything. They are everywhere. They will come up to your knees, it's everywhere. And they are extremely important for nitrogen cycling and carbon cycling in all of these areas because they're a major, they're just everywhere, like I said multiple times. So I went to Toolik Lake, Alaska to get samples of these mosses. This is a map of sort of where we went. We collected samples in between Fairbanks and Toolik Lake Station. The samples that I cultured from specifically are from Toolik Lake, so from above the Arctic Circle in the tundra. This is some pictures of the kinds of areas where we would collect samples from. So you got foresty areas, you got tundra areas. This is the last spruce tree that we found. This is really kind of the areas that my mosses are specifically from, tundra areas. This is Toolik Lake. So it's really exciting going to Alaska. We have a lot of adventures, to say the least. It was a lot of fun going through the trials and tribulations of dealing with your car overheating on one of the most dangerous highways in the United States in between two forest fires. So it was great. But it was a lot of fun. It was very pretty, and there were a lot of blueberries. They let you pick them, and I loved them. So I collected mosses, but what specific mosses did I collect? It's a great question, audience. I collected Tilium, Polytrichum, Dicranum, Hyliconium, Sphagnum, and Aliconium. I did cultures from all 6 of these. I have a lot of cultures at the end of this. So I'm going to relate the boreal mosses I just explained to you to the microbial communities that I ended up filtering. So we have your boreal mosses, and we take those, and those are in conjunction with a lot of communities of microorganisms. And it's really important to understand the function of the mosses with microorganisms because together they kind of help us understand more about the environment. And a lot of these microorganisms have really unique functions, such as like the nitrogen cycling, that we need to understand. So culturing them is super important to better understand how all of this works and how this is going to affect the future of climate change in northern areas. So my goal for this project was to grow a broad variety of uncultivated and functionally interesting groups of microorganisms. So I started by culturing them. I took moss slurry and sort of spread it over plates and got a wide variety of things. I did this culturing them in a variety of lights and media types, and eventually isolated them so that hopefully each one would be on their own plate. I then sequenced their DNA. They have a specific barcode gene that you sequence. And I used that to kind of determine who was who in all of this data. This culminated into my very pretty phylogenetic tree, which is the relative relatedness of everything I cultured to each other. So everything with the little red dot is something that I grew in the lab. I had some very, very interesting things. I actually had 2 novel species. This first one is Nucella agnibacter. It has a 94% identity with its most closely known relative. And that may seem kind of high, but you have to realize that we're like 99% related to chimpanzees, and it's really similar with bacteria. So I also cultivated Frondi habitans, which is 93% identity. I think that one's really cool because it seems to be very uncommon. It has only a few known species right now, so that's a really interesting one. I grew a few other cool things too, especially this purple one here, which has antifungal properties and is used by scientists to help with preventing amphibians from dying around the world from fungal infections. So, moral of the story is I cultured 13 unique genera from 4 different phyla. The genus Pseudomonas was the most abundantly cultivated. I cultivated 63 out of 89 of my successful cultures. And many of these cultivated microbes were cycrotolerant and had antifungal properties. Cycrotolerant means that they like to live in cold or are okay with living in the cold. So future direction, cultivation of a broad diversity of interesting bacteria would likely be achieved if I did this longer. A lot of bacteria, especially probably in cold northern environments, take a really long time to grow, taking months, only had 9 weeks, so probably do that. I'd like to acknowledge pretty much everybody, the RECCS program, Beer Lab, Hannah especially. Yeah, you guys, everybody was super supportive, and I appreciate it. Bibliography.
Yeah, I'm just wondering about your field collection. So were you just kind of identifying the moss and then just taking a sample, labeling it, that moss, where and what it was, and then figuring out what it was back in the lab? That's what you were doing?
Pretty much, yeah. Like we just identify them in the field, so. Yes.
You mentioned incubating with different light conditions.
Yes.
But most of these Pseudomonas and other ones don't actually use light. Do you find some that are bulletproof?
That's a great question. I did not and could not find any metabolic information that would suggest that I cultured anything autotrophic, but I did try to, for sure. Yes.
Are there any interactions between moss and fungi that you know? A lot of the things you filter properties maybe like types of moss.
I don't know for certain, but I can speculate since the north is pretty wet that they would probably have some pretty significant interactions with fungi. I know that my plates, I had a lot of fungi that grew on them, so maybe the bacteria and their antifungal properties help with dealing with that on the mosses, but I can't say for certain on that. Yes.
Given sort of the diverse range of conditions in which these microbes probably like to grow, how did you choose the media that you were going to test out in the facility?
One of them was a really kind of wide-ranging media. It was R2A. I don't know who's familiar with that at all. So that was the one that was kind of like gonna catch a lot more stuff. I also used an AP media, which is pore-growing and oxygenic phototropes, in the hopes of getting something on that end too. But really, like, I just chose whatever worked and was available.
What is about your microbes with the moss?
I wouldn't know, but
Because you grew them in different media, isolate them entirely from the moss.
yeah, I was for the ones that I used. I only had a couple that I did not have complete isolates of, and I don't know which ones those are off the top of my head. I didn't have enough time to go through and find out which ones didn't want to isolate from each other. That's a great question.
I can see there's a lot of experts in the audience currently on our topic. So yes, we can continue this at the end, of course, but let's thank And our next speaker, who is the second to final speaker, will be Haley Nolan. And at around 3:30, we will close with a quick ceremony, acknowledgement, and reception and question survey team close to campus here, and her primary mentors were Jen Underwood and John Moody. Go ahead, Moody.
Hello. So my project for the summer was modeling downstream changes, a Lagrangian study of sediments and microbial communities along the Powder River in Wyoming and Montana. So sediment is considered one of the #1 pollutants in America, and with it, it carries many other contaminants such as helpful and harmful bacteria. Sediment accumulation also leads to environmental problems such as erosion and affects aquatic ecosystems, which is why it is important to study this to predict and track the flow of sediment and its associated biological constituents. And in order to do this, we have to look at the relationship between the flow of the river, the transport of sediments, and the occurrence and distribution of the microbes. Bacteria is present in a few different parts of the river. One is free-flowing in the water column. Another is that it's attached to sediment particles in the river, suspended sediment particles. And then also it is in the sediment and rocks up in the riverbed. So our research goal was to measure the changes in the transport of sediments and microbial concentrations as water travels downstream through the Powder River watershed. So the Powder River resides in Wyoming and Montana, and we chose this river because it carries very high sediment loads, and so it's a good reference river to study the transport of sediment. So we measured the sites at we measured things at 9 different sites along the Powder River by following 2 water masses. So what I mean by 2 water masses is that we use a Lagrangian method of sampling. And so this means that we followed the river and sampled as we followed. And so theoretically, we would be sampling the same water mass at each site. So there's 2 different ones because right there after that purple star, the river really meanders a lot. And so we would have had to wait days to catch that water. So for time constraints, we ended up having a second water mass over here. So each site varies by geological formations and the flow of the river, how wide the river is. So here's some examples of the sites we were at. This is Clear Creek confluence where it meets with the Powder River, which is really crazy because it's such a stark difference right there. So at each site, we measured the discharge of the river by measuring the depth. So this one is if you're facing down the river, your left is the left bank, and it kind of shows by measuring the depth, the shape of the river at the bottom. And we use this to estimate the area by taking parts like this and estimating what that area would be and then finding the total. We also took velocity measurements at parts of the river, and then we used these, multiplied the area by the velocity, and we got the discharge or how much water was flowing through the river per day. So at each site, we also collected sediment using these lovely buckets and 63 micron sieves. We collected sand, which is above 63 microns, and silt, which is below. We also collected DNA from the water by filtering it through the RECCS filter. And then also after the sediment settled in these buckets for 2 hours, we also filtered water through there to collect the DNA on the RECCS filter. That's called planktonic, and it's the microbes that are present after they have separated from the sediment that they were attached to. So then we analyzed this in the lab. We found the dry weight concentration and in silt. From the same silt and the 2 water samples to find how much biomass was in either per liter of water or per percent. And then we calculated.
I can try. Would you do the rest of your research?
And then
I can put your computer in your lap if you want.
times the concentration.
We can do it.
Exactly how much biomass and sediment
It's not her yet.
So this is, with the floods, there's a lot going on here, but the yellow line is the discharge or how much water was flowing. Ones that are circled are the tributaries, and then the ones that are not are actually the Powder River, because we sampled at sites either at tributaries before they entered the Powder River or right after to see the changes. And then these lines are the silt and the sand, how much is being carried, and then all of the bars are the biomass of the different things we're looking at. So as you can see, so right here, they're all pretty similar, which kind of verifies that we did a good job doing a Lagrangian study, because since we're taking samples of the same water mass, it's a good thing that they're pretty similar. And then also as the sand and silt concentrations go up, so does the biomass, which is a common trend we saw. Also, it gets higher at the end as the river gets wider and tributaries are entering into it. So this is the concentration of each of those. It's kind of set up the same way. And so this one is a little bit more variable. It changes per site, which was interesting because there's different types of biomass that are more present at different sites. One in particular that is interesting is Salt Creek right here. It's a tributary and it's known for high conductivity, and the water is pretty salty. Like I had to siphon the water off of some of those buckets and you can taste the salt. But what's pretty crazy is that there's high biomass in the water there, which we don't know why. So because of this variability, it calls for further testing on other factors, biological and physical, that may be affecting how much biomass is at each site. So I just went over all of this. Some limitations of this were that, so all the data that we took was at one time point. And so while this gives us a good baseline, we do need more information to gain a better understanding. There was some sites with limited sand or silt, which was another one. And then future studies will focus on the composition of the sediment and the microbes and the chemistry of the water. So if we can identify who's there, what they're doing, and identify exactly what there is, we can better track contaminants and figure out at the source how far they're traveling. These are my acknowledgements, and then I also have some pictures of our field work that was well. He joined us on the field one day. So there's a lot of things that affect the discharge or the flow of the river, and that is including seasonal things. So later in the summer, there will be less discharge, which I think there would be less things being carried by the river, and there's a lot of things that go into it, but that's the basic.
Hannah and then Nick.
Do you have any idea of how much, like in your water mass, how much kind of mixing happens in depth as it is traveling the river? Like, is it the top layer of water pretty stable all the time, or does it?
So it does mix. It is important for things like velocity when we're measuring to go to, it's actually 6/10 of the depth, because the water does move at different speeds throughout the river. So at the top, it's moving more quickly, and then at the bottom, as it's catching rocks and stuff, it moves more slowly. And so about the average, like a good mixture of the water, what I would assume would also be about 6/10 of the way down.
And Rick, really quick, maybe as, yes, really quick.
So this follows on that. So how did you know that you were sampling the same water bodies?
So you can go through and like time it by the velocity, and which we did later to check. We were sampling about the same, and then I think we would have had to wait like 4 days in between those two. So.
All right, thank you so much. Right, our final speaker is Nicole, and she's going to cold and chilly.
But warm on the inside.
Warm on the inside, exactly. Nicole also worked up at the long-term ecological research site at Niwot Ridge, and her mentors on this project were Kate Hale.
These white triangles is where we took manual depth measurements, and at the darker spots is where we took density or sweep points. So, to get to the depth, we took measurements with a probe, which is a fancy word for a big old ruler. And so we also got density by doing snow pits, and so we would dig down to the surface, and you would take cuts out of the snow, weigh those, divide that by the density of water, and then multiply that by the depth, and you get sweep. We would also use federal samplers, which would give you sweep directly by weighing the snow after you stuck it in the ground there. So a little bit about fieldwork experience. It's pretty unpredictable, and this is my first time going out and diving into something this high up, really. And so there were some times where we were chased out by storms. We also got to dig a 10-foot snow pit. That's me at the bottom there. And we also, this is the Tundra Lab. We got to spend the night up there and watch the sunset, which was amazing. And my team was just a great conversationalist. They were great people to be up there on the ridge. So what did we find? So the upper snow depths are very uniform compared to the lower snow depths, and you can see that by with the standard deviation that the lower catchment varied more than the upper catchment. And so here are the averages compared, and you can see that the lower snow depths was consistently higher than the upper. And so here's some melt rates that we have going on. This is weekly melt rates in between the times that we went up there, so we took measurements every week. It snowed in the middle of June, so you can see how that affected the melt rates there. Kind of odd, but you know, it happens. Again, the lower catchment was consistently higher melt rates. And right around here is where the storm happened as well, so you can see how that affected the snow water equivalent. Again, lower catchment consistently higher. And so some significance of the study is that we found that the more variation with slope and aspect meant that there was consistently higher depth rates, melt rates, and ultimately water available in the snow. You would expect to find you'd expect to find deeper snow at higher elevations, and that's not what we found, most likely because the upper catchment was a very exposed face, and so wind disposition and scour would be moving the snow off of that higher elevation. Also, this is about a 200% above average snow year. You've heard with the pika and the flowering times that the snow lasted much longer than anticipated. And lastly, some other factors that can contribute to snowmelt distribution is vegetation, wind scour and disposition, and temperature. Yeah, that's that.
Great, questions for Nicole? We're back on schedule. Yeah.
Beneath your slides, you have small, which snow versus depth.
Snow what?
Snow what you put it, SWE.
Yeah.
Versus depth, and that wasn't a linear plot.
Yeah, that was just more showing the relationship. Oh, we're just going right on through. Yeah, that was more showing the relationship between snow depth and snow water equivalent. So generally, just as depth increases, so does snow water equivalent. As it decreases, so does the snow water.
It comes back down, which is where.
Yeah, that's just showing that typically you have depths that you have a rise in depths, and then it's just more showing the relationship. It's not a specific like depth circle back around necessarily, but yeah.
Any other questions? Yeah, Nelson.
I did a paper on
Yeah, definitely. So most of the interns here were in Boulder, and there was quite a bit of flooding and a lot of rushing water that hadn't been in the past few years. So if this was again an odd snow year, and so the trends over the past decade or so has been a lot less snow. So if you do have more snow, that is going to affect like reservoirs and just water availability in general.
I have a quick question. I love seeing some of the field work in action shots that you showed. Was it freezing cold up there? This is not a very scientific question, but
yeah,
it looked pretty warm.
Yeah. Actually, in the last slide here, I had a picture of Siobhan, a team member, myself, and Kevin, another team member, and I am bundled up, like completely head to toe, ready to put on goggles. Like, I personally found that it was freezing. But people who are used to the snow up there were ready to go, like ripping off layers once we hit the snow. And I would say you kind of get used to it, but yeah, it's a lot colder if you're not used to it, getting up to that elevation. So,
and
yeah.
How did it change when you were at the bottom of that deep pit?
It was actually a little bit warmer than I thought it was going to be because the snow is pretty isolating. And so, especially when you're digging 10 feet down, warms you up for sure.
So any other questions, scientific or not so scientific? Yeah.
Just a logistics question. Did you find like a great tactic for carrying around that? Like what worked?
Yeah. No. So there was a few times where I could use a smaller one if the depths weren't as great, where you could just fold it up nice and neat. But the other one was a long pole that you had to screw together, and so I would just trek out there with my giant pole. And one about like the first or second time that I had gone up there, there was a giant snowstorm that came in and there was lightning, and so I'm running around on the top of the mountain with a giant metal pole. But you get used to it, and then you learn to appreciate it. And once you're carrying up other heavy equipment like a ground penetrating radar, that becomes easy. You're like, oh, it's just a pole, no biggie. Or that federal sampler, that's a ton of tubes packed together on like this backpack, I guess is what they refer to it as. But that is also really heavy. And you can slide a lot on the snow. I mean, there's a lot of ice, and so. Oh yeah. Yeah. It was great. It was so much fun. Yeah.
Well, great job describing.